pretty much the entire course, which is a course about perception, I don't want to say ignored, but I focused primarily on vision. And that's partly because, we are, visual animals. I think it's hard to imagine the world without vision. And it's such a kind of prominent and kind of a center front kind of sense. At the same time, we can't really imagine ourselves without the other senses, let alone without hearing. So hearing, one kind of very, very interesting feature about it is that it's always around us. So in contrast with vision, you can always close your eyes. Or alternatively, when it's dark, right? I mean, you can't see anything. Or, I can, blind you with a strong kind of light. And it's really hard to turn off your hearing. You can't just not hear, right? And so I think that's one of the most, marvelous things about this sense. So the idea that it's always there, it's an extremely, extremely, extremely sensitive sense. So you could really sense things that you wouldn't think are imaginable, and I kind of talk about that as we go along today. So overall, this function of hearing, which we are going to call audition, okay, is extremely important for our survival. So imagine, when you're sleeping and, something happens and you can't hear that, or imagine that you're walking in the forest and someone, or some animal creeps up kind of behind you and you can't hear it. So obviously, the function of hearing is extremely important, extremely significant in terms of evolutionary kind of consideration. Let me just kind of make sure that we're on the same page in terms of what those terms are. So again, I'm talking about audition, okay? Which is again, a fancy word for, and let me change that, which is a fancy way of saying hearing. And the other thing we're going to talk about today is the auditory system. So the same way that we talk about the visual system, we can talk about the auditory system. Okay, so the visual system is the system that's responsible for processing information coming through, the visual sense, one would say from the eye, although that's also in dispute, but I'm not going to go into the deep philosophical kind of roots of that discussion. And the auditory system or audition is everything that's going to be related to hearing. We're going to talk about the basic properties of sound. What is sound? The same way that we've discussed light and its nature, if you recall. And then I'm going to spend, a substantial amount of time today talking about the structure of the mammalian auditory system. Now, I will primarily be talking about the human auditory system, but in the beginning, it's kind of important to compare our auditory system with the relatives, which are other members of the mammalian species. Okay, so that's going to be very important. And then if we'll have time, we will be talking about some kind of operating characteristics, or what are the properties of the auditory system and how sensitive we are to different features of sound in the environment. So let's talk for a little bit about sound and what is sound. Okay, so for those of you who might have heard about that or maybe have a little bit of sense of that. So sounds are created when objects vibrate, which is kind of interesting. Remember, when we talk about a sensory system or about the perceptual system, we talk about a physiological signal that is transducing some sort of a physical signal. Okay, so a change in energy, so we have to translate the world around us. Here it's going to be about vibrations that cause molecules in the air, and we'll talk about other media in a second. Those would create pressure, okay, and changes in pressure that would reach our ears. So sound, maybe I should start with that, kind of, disambiguating things. Sound comes from pressure fluctuations in the air. Now, you may be, that may sound confusing a little bit. What do I mean by pressure fluctuations in the air? Is this like barometric pressure? Is this weather? What am I talking about here? So the idea is, if you think about it, let's say if you take a tuning fork and you hit it, right? That tuning fork creates, vibration. It vibrates. That vibration in the object creates vibration in the medium that it's adjacent to. That medium, is called air. Okay? So object vibration, in this case the tuning fork, and I'm sure that you've all seen a tuning fork, I don't know, in the cartoons or maybe in real life. It's this kind of object that looks like that. Okay? And it produces what we call pure tones. So tones that are in a single frequency. So you hit this tuning fork like this right here, and then there's going to be a kind of a sound that's going to emanate, look at that, from that tuning fork. Okay? Now, those lines, basically what they refer to, what they kind of signify, is those air molecules that are also going to vibrate. Now, eventually, they're going to hit your ear, okay? And then those changes in pressure, okay, because it's going to hit your ear, right? So that's going to create pressure on your ear. Those changes in pressure are going to kind of be transduced into a neural signal that is eventually going to be known as sound, or the perception of sound. And we can plot these vibrations or these variations in pressure on a graph, and those would look something like that. Okay? So if here's a speaker, so they're using a speaker, I was using the example of a tuning fork, but you'll see the tuning fork in 2 slides from something like that. And so the speaker, I don't know, has anyone here, ever laid a hand or kind of stood close by to a speaker? So if you can do it, after a class, if you just put your hand on the speaker and you let you operate the speaker, so you'll get this feeling, this kind of tingling feeling of vibrations on your hand. What it is basically, what's inside the speaker is an actual membrane. That membrane is vibrating, okay, as a function of different electrical pulses created by or recorded by, whatever, the CD that you're hearing, or back in the day in the record. So the sound is creating vibrations, which you could pick up with your hand, but you can also definitely pick up with your ears. Those vibrations or changes in pressure are plotted here in B. Okay, so on the y-axis you have increase or decrease to air pressure, where you can think about this baseline here, this dashed line as a baseline, which is just your normal atmospheric pressure. So this is kind of related to weather to some extent, okay? And so any change from that is going to be considered as a sound, okay? So we can plot it as a sinusoidal wave, okay? And we've mentioned those waves, when we talked about, for example, about light, and how it can be both a photon and a wave as well, right? And this would be the distance from the source, and in that case, it's also going to be translating in time, okay? So a lot of the plots will be not necessarily in distance, but also in terms of time. And if you're following so far, how do we call units or fluctuations in a wave over a restricted period of time? How do we call that? So the number of fluctuations, or the numbers of ups and downs from peak to trough, how do we call that over time? So that's going to be known as the frequency of that sound. Okay, so just to recap, sound comes from pressure fluctuations in the air. And when such pressure variations are plotted on a graph, they form a sine wave. Okay, so far so good. Now, if it's a sine wave, that means we can describe it in at least 2 ways. There's actually a third one which I'm not mentioning here, but the first or two, or the cardinal 2 in our discussion, are going to be 1, its amplitude, and 2, its frequency. So I'm trying to describe now, okay, the sine wave that I've just shown you right here. So let's talk first about amplitude or intensity. Amplitude or intensity is the magnitude, okay, of that displacement, that increase or decrease of the sound pressure wave. Okay, so how far up and down you're going to go. Let me see if I have an example here. I do, so let me just quickly jump to that slide. Hold on one second. So what I'm going to do now is I'm going to demonstrate to you changes in, or what is amplitude, okay? So amplitude is basically this height, okay? From peak to trough. So in this case, for example, we have again a tuning fork, and now we're going to hit it stronger, okay? And when you're going to hit it stronger, it's going to create greater amplitude. So the wave is now, you can see that its height becomes greater, and then we can say that its amplitude in this example has doubled. And I'm not showing you, but this is in comparison to an original one, which looks, which looks something like that. Okay, so here you have a certain vibrating body, in this case a tuning fork, okay, and then it grows in amplitude. So hold on. Okay, amplitude here is doubled. Now that's one feature of the wave. And if you remember that we talked about that when we talked about, spatial frequency, if you recall, we talked about those. And now we can also talk about frequency. And frequency is just how many repetitions, how many times per second you have a pattern of pressure change. Okay, so we've already mentioned that. I just want to kind of show you. So here, you have greater frequency. So it's the same amplitude, but just double the frequency compared to this thing, which I'll show you in a second. Okay, so long story short, the number of cycles in a second, that is going to be defined as frequency, and frequency is going to be measured in hertzes. Okay, now why is it so important? Why are we describing, amplitude and frequency and talking about waves? I mean, this is not a physics class, right? The reason for that is because those key dimensions of the sound wave are actually translated or have a direct translation in terms of their subjective experience, in terms of their perceptual experience. So the amplitude of that sound wave, that's going to be translated, or it's going to be perceived as loudness, just how loud that sound is. Similarly, frequency is going to be perceived as pitch, just how high that sound is. Now, sometimes, especially in everyday kind of, discussion, we tend to mix the two, but we should really kind of, distinguish between loudness, okay, and pitch, and it's not the same thing. So loudness is just basically how much you're going to tune your volume knob. Does that make sense, right? So how loud is that sound? Whereas pitch, which is based on frequency, is going to be the just how high or low that a particular sound is. So I'll give you an example, right? So if you're going to listen to a tuba, that's going to have a lower frequency compared to, say, a flute or a clarinet. And irrespective of that, you can always, increase the volume up or down. Okay? So loudness and frequency, loudness and pitch, that's the better term, are two independent perceptual dimensions. Does that make sense? So they're based on two independent physical dimensions, which is the intensity and the frequency, the amplitude and the frequency. And they can be translated, and I can show you that here, into loudness or pitch. So again, just to kind of recap and having, if you guys are taking notes, so loudness is the psychological aspect of sound related to perceived intensity, and pitch is the psychological aspect of sound related to the perceived frequency. In terms of units of measurement, how do we measure amplitude and frequency? So frequency I've already mentioned in a way, so let's just mention that again. So frequency is basically how many cycles you have in a second. And so 1 hertz equals 1 cycle per second. Okay, so let's imagine, for instance, let's say here, and let's say that, I don't know, from this point to that point, that's a second. Okay, or from that to that point. And then you're going to have 1, 2, 3, 4, 5, 6 fluctuations in a second, which would be a sound on the frequency of 6 hertz. Okay, so 6 cycles per second. Now, I don't know if anyone here still listens to the radio, anything, beyond Spotify and Pandora or whatever, music app you're listening to. But if you look in the car, or, at home, and you're going to tune your radio, you're going to see some numbers on it, right? So let's say, I don't know, what's your favorite radio station? So let's say you're going to look at, I don't know, Mix 1077. That's one of my kids' favorite radio station. So what you'll see is that it's basically 107.7. But like all measurements, we can't have a value without the actual measure itself. And what you'll see is megahertz. Okay? So that means that you have a sound, or that you have a certain range within the sound range that we're sensitive to, and we'll describe that as well. That is at the wavelength, if you'd like. That's the analogy, right? We talked about wavelengths in vision. That is at the wavelength or frequency of 107.7 megahertz. So does anyone know what mega is? It's a million. So basically, in terms of the wavelength, in terms of that frequency of that radio station that you're listening to, it's broadcasting at a speed, quote unquote, or at a frequency of 107 and 700,000 hertz. Okay? Does that make sense? So 1077 would basically mean 107 million. 0.7 would be 700,000 hertz. Okay, so that's pretty high, right? So that's important to remember. That's the unit of measurement for hertz. And I keep going up and down. And then for intensity, and this is a very important and interesting unit of measurement, we're going to measure that if you've heard the term decibel. Okay, so that's a unit of measure for the physical intensity of sound. Okay. Now, in contrast to hertz, which is an absolute measure in the sense that, you have 1, 2, 100, 1,000, 1 million, cycles per second, decibels are actually measures which are known as, relative measures. So they basically describe, and I know it's going to sound a little bit weird, so bear with me. They define the difference between two sounds as the ratio between two sound pressures. So there's going to be a certain baseline or a certain kind of unit that we're going to kind of agree upon. Okay, so we're going to call that the reference pressure. And then basically whatever you're measuring is going to be measured compared with that. That ratio is going to be known as decibel. Okay, or specifically it's going to be known as SPL, or sound pressure level. So sound pressure level, as the name kind of denotes, is the ratio between what you're actually measuring and that reference pressure. And this is defined as, I'm just looking at the book here, it's 0.0002 dyne, which is a unit of pressure, of physical pressure, per square centimeter. And we'll mention that in a second when we talk about the actual scale and how we measure that thing. Okay? Just as an example, before I'm actually going to explain that logarithmic scale, the idea is that, for instance, if you have a certain sound that's about 10 times more pressure than your reference one, okay, that's going to be equal to 20 decibels. And I'll show you the formula later today, but you just need to understand that it's the ratio. And therefore, 100 over 1 is going to be equal 40. Now, why is that? I mean, obviously, 100 is, 10 times is 10. But if you actually think about the power, okay, so 10, right, is 10 to the 1. And so that would be, so we're looking at the power. Okay, that's going to be translated into 20 decibels. And then if we're going to look at 100, that's going to be 100, that's going to be 10 to the power of 2. And that's basically going to be 2 times that, which is 40. And if you don't understand it yet, that's fine. We'll explain that as well. But long story short, decibels is a relative unit of pressure. And if you think about it closely, there are other measurements that you know without realizing that they're also relative measurements, and things like temperature. So for instance, let's forget about Fahrenheit. If we talk about Celsius, right, 0, that's temperature of freezing. And then based on that, you're going to add more and more degrees. Okay? And of course, 100 Celsius is the point of boiling. And of course, that can be translated to Fahrenheit, and et cetera, et cetera. But the idea is that it's relative. So when you're measuring, let's say, 10 Celsius degrees, okay, so that's 10 more than the point of freezing. That's your reference point. And again, loudness is the psychological aspect of sound related to intensity. So intensity, the physical, is going to be the physical the physical dimension is going to be measured in decibels. Pitch is going to be measured in, pitch is the subjective perception of frequency, which is measured in hertzes. Okay, and again, low frequency correspond to low pitches, like the tuba I was mentioning, and high frequency sounds correspond to high pitches. Okay, so again, just to demonstrate that, imagine that you have a tuning fork that's going to create compression and expansion, right? Those are the changes in pressure of air molecules produced by the tuning fork's vibration. Okay, so you can think about having, basically having these molecules kind of bounce, back and forth, compressing and expanding, compressing and expanding. Okay, and those are described again by amplitude. Okay, and by a frequency. So here is amplitude and here is frequency. And what we do in the psychophysical realm, in the world of psychoacoustics, we can plot the people's sensitivity to both dimensions. How do we do that? We plot these kind of things. Okay, so what you see here is the range of human hearing, which describes how much we're sensitive to both different frequencies and different intensity levels. And you can see them here. So on the y-axis, you have the sound pressure level. Again, that's intensity. So this is the physical property of the stimulus of amplitude or intensity. And here's another one, that's frequency. Okay, so imagine again that I'm going to use some of the psychophysical method we've described in the first couple of weeks of our course. And I'm going to measure your sensitivity across each one of these conditions. So you can go to the acoustic booth, and I'm going to basically project to you or present you with different sounds. They might vary in frequency, they might vary in intensity, etc., etc. And I'm going to record if you capture those or if you are sensitive to those stimuli or not. And what you can see here, and for those of you who remember, the lower you are, the more sensitive your hearing is. In other words, you can hear more, or you can hear, how would I put it? You can hear sounds at a fainter and fainter level. So the more sensitive you are, the lowest this plot is going to look like. And if you recall, then that allows me to say, plot kind of a developmental, change where I can see whether people get better or worse over time. Then I can also look, because this is frequency by intensity, I can see whether that shape, this kind of inverted, or not inverted, but this kind of U shape, is going to change over time, and does it change differently for men and women? And it's kind of ironic because men, over time, they're losing sensitivity to distinguish high frequencies. So in this plot, and I hope you're kind of following what I'm describing here. And so in this kind of plot, let's imagine that you're a guy and you're aging. So yours is going to look maybe something, let's say, like that. So beforehand, you were sensitive to higher frequencies. Okay, what do I mean more sensitive to higher frequencies? In the sense that this frequency could have been detected at a much lower amplitude. Okay, now you need much higher amplitude in order to detect the same frequency that you've sensed before. Does that make sense? So let's imagine that I'm going to compare my ability. Let's say that this is when I was 20. This is when I'm 50. Okay, this red line. And let's say I'm going to look here at 2000 hertz. Okay, so beforehand, I needed only 10 decibels in order to detect a 2000 hertz frequency. Now I need to amp it up. I need to reach, I don't know, let's say about 40 hertz in order to actually hear that sound. Okay, so that's loss of sensitivity. Now, ironically, and I don't know if, nature plays kind of cruel tricks on us, women actually show a different trajectory in terms of, aging. And what they show is the following. Let me just pick a different color here. Let's say, whatever, blue, or maybe purple. So women actually have a less sensitivity in the lower frequencies. So theirs looks, sorry, I should have done it differently. Hold on, let me just erase it real quick. Why can't I see the eraser? Okay, I can do it differently. So women basically now, they have a different trajectory in terms of their aging, which looks something like that. So they're actually less sensitive on that direction. On the low frequencies. Come on, pencil. So a low sound that they might have heard, better, let's say when they were 20, now they need much more in order to actually detect it. Now, of course, and that's the great irony here, men's voices are actually in the more lower frequencies and women's voices are more in the higher frequencies. So that means that over time, men and women find it harder and harder, physically speaking, okay, not psychologically, and to hear each other. So that's kind of an interesting joke. But most importantly here, the idea is that we as species, we're extremely sensitive. We can hear frequencies that range from 20 to 20,000 hertzes. Okay, so that's, a scale or 4 orders of magnitude different. And importantly, it's going to be across a wide range of intensities. Okay, from, by the way, look at the minus here, but that's maybe something for those interested listeners. How is it possible that I have a minus? But you can ask me that, after class. But look at the range. So it's from 0 all the way up to, let's say, 110 decibels. Now, remember, this is a logarithmic scale, which means we're talking here about orders of magnitude. Okay? And importantly, you can see that at a certain point, then you're going to have a high-risk threshold, meaning that, you're going to pop your ears. And then at another range, when it's too loud, you're basically going to be in pain. And I don't know if you've ever experienced acoustic pain, but it's pretty painful, pretty painful. Okay, but importantly, just I want you guys to kind of get used to looking at these kind of plots, and you can basically kind of plot the ranges that are kind of, again, in terms of the physical properties of the stimulus, and kind of where they are in terms of the range. So here is speech. So most of our speech, and of course it also depends on different languages, lies around this kind of spectrum, okay, between, let's say, 200 to, say, 10,000 hertz, and then at a certain intensity. Again, kind of usual standard speech. You can see how music has a much wider range. And let's say the non-specific sounds that may not necessarily have certain meaning, you can see that there is a much broader range. Does that make sense? So we can detect voices, or sorry, I could put it this way, we can detect voices at a much narrower range compared to musical instruments, and that's even much narrower compared to the broadest audible range. Okay, does that make sense? That's an important point here. Okay, and again, those sensitivity functions which change with age, we're going to call them acoustic sensitivity functions, right? Okay, so now, again, let's talk a little bit about those decibels and those, sound pressure levels. So if you looked at that range carefully, you saw that there is a ratio between the faintest and loudest that can actually range up to 1 million. And, well, I mean, you have to understand, what those decibels stand for. But the idea is that because we are sensitive to such a wide range of intensities, you have to figure out a way of putting everything in the same scale. And I don't know if you remember when we talked about that, when we talked about the visible range of the spectrum. And if you remember, I presented a scale, and that scale was in a logarithmic scale, because that allows me to compress the actual range. Okay? Because imagine that I want to present, 1 centimeter and 1 meter in the same figure. That's obviously very hard when I'm looking at logarithmic units. And logarithmic units, again, they're units of power. So an increase of 1 is basically an increase of the power. Okay, I hope you remember that. But long story short, that's the same thing with decibels and with intensity. And so we can compress everything by describing those differences in amplitude in a logarithmic scale. Okay? Now that's important to realize, because relatively small decibel changes, and you may think, oh, it's just whatever, it's just, 5 decibels, can actually correspond to huge changes in terms of physical changes in the environment. Okay? So if I'm going to talk about, let's say, an increase in 6 decibels, that would correspond to a doubling of the amount of pressure. And you can actually see it in this plot, where we can see that the sounds in our daily life, they vary greatly in their intensity. And you can see it here, right? And so, a change, let's say, let me see if I can give you an example here. So between, let's say, a heavy truck and a jet, so between this guy and that guy, that's about a 44 decibel difference. Okay? But if you translate that logarithmic scale into the actual difference, in terms of intensity, the jet is actually almost 158 times more loud or louder than the truck. So those 40 decibels seem innocuous enough, but again, they would translate into something that's louder, way, way, way louder than what that scale would actually kind of tell you. Okay, so those were kind of the two kind of main things I want you to take from this loudness, which is basically our way of perceiving intensity, and the pitch, which is our way of perceiving frequency. Okay, so now the question is, well, how are those sounds, in terms of those two dimensions, are actually detected and recognized by the auditory system? And it's important to realize, because audition is such a fundamental sensory modality, such a fundamental sense, that it evolved over millions of years, and we can actually compare different animals because of different environmental constraints and different kind of requirements, and how they've evolved to develop different, auditory systems. So many animals have very, very different hearing capabilities. And, the example everyone gives in this case is always dogs, right? So dogs can hear higher frequency sounds, but no one mentions the elephants. And elephants can actually hear much lower frequency sounds than human can. And you can think about that because, elephants, right, are heavier. And so that would translate into lower frequencies. Elephants, it's very important for them to recognize the sounds of their mates from a distance where they can't see them necessarily. And so it's very important for them to be sensitive to those lower frequencies because lower frequencies also travel longer. Okay? Or maybe I should say, high intensity, low frequency. So high intensity, imagine that, an elephant walks. I'm sure you're all familiar with the Elephant Walk song or tune. But when it walks, it's basically creating very, very strong vibrations, which again translate into low frequencies, which basically kind of go, way longer than your very high kind of chirps, let's say, of birds in terms of high frequency, or maybe even dogs. Okay, so let's talk a little bit about the actual physical or physiological mechanism that allows us to perceive sound. That's obviously, or at least part of it, is the ear. And I didn't put numbers here, but we're going to talk about different parts of the ear. Basically, we're going to talk about the outer ear, right? I think you're all kind of familiar with that, I would hope. And then we're going to talk about the middle ear. And then we're going to talk about the inner ear. So remember those kind of perceptual journeys I was telling you about. So we're going to follow the sound, and we're going to follow its progression from the ear, or from the outer part of the ear, all the way through the inner ear. And then we're going to talk about the actual neural pathway, okay, that goes and ascends all the way up to your primary auditory cortex, to your A1 cortex. Okay, so from sound in the environment through the ear and its different parts, the outer, the middle, and the inner ear, and then it's going to be transduced into a neural signal that's going to ascend up the auditory pathway all the way up to your primary auditory cortex. Okay. So let's introduce this thing here. How do you call that? You probably call that an ear, right? And so you're partially accurate. So we're going to talk about what we would call here and you can see it here, the pinna. And plural, that's pinnae. Okay? So the pinna is basically this device which allows you to amplify the voice. And you can see that different animals, right, have different shapes of pinnae, okay, or ears. Because the reason why there's a difference, because that basically describes like this kind of dome shape of the ear. But the ear itself is comprised of all the machinery inside it as well. That's why we don't use the term ear. But of course, in everyday language, when you talk about an ear, you're basically talking about the pinna. And of course, different animals have different sizes, different shapes, as well as different abilities to move their ears or their pinnae. And that is to localize or to be more sensitive to different sources of sound. So the idea is that the length and the shape of the ear and the ear canal within it, they would enhance certain sound frequencies. Okay? So, I think it's really marvelous just to look at this kind of variance and see how different environmental pressures leads a certain organism to very different solutions, although the principle is, pretty much the same. Okay, so in the outer ear, as I've already said, we have the pinna here. You can see it here. And then we have the ear canal. And we're going to call that the external ear canal. And the purpose of the ear canal is, first and foremost, is to kind of insulate, I'm going to start with that, and protect the main mechanism, or this kind of thin layer, which we call the eardrum, or the tympanic membrane. And we're going to protect it by having this kind of secret passageway into it. So, when you're using those how do you call them, those sticks to clean your ears, this is why it's important not to stick them, too deep, because you might actually hurt I mean, yeah, usually you don't, but you might hurt the actual eardrum or the tympanic membrane, which is the first part, introducing the sound into something that the brain can work with. Okay, so that's the ear canal. But more than that, the ear canal, in conjunction with the pinna, would actually collect the sound waves and funnel them to that tympanic membrane. Okay, so again, I think that the notion of a funnel is very apt. Or if you think about it in the other way, when you're using a funnel, like in one of those 19th century loudspeakers, you can understand what's being done, but in the opposite way, because basically that magnifies the voice, right? So in the same way, that would kind of take that voice or that sound wave and funnel it into this mechanism here, and maybe zoom on it, and that's going to be your tympanic membrane. Okay, so basically, just to kind of recap, the outer ear is kind of the pinna here and the ear canal, and that leads to the eardrum. Okay, so the tympanic membrane is kind of the border between the outer ear and the middle ear. So that's everything behind it, and we'll talk about that in a second. So let's talk a little bit about the tympanic membrane first. Sorry. Here we go. So the tympanic membrane, or the eardrum, as you probably are familiar with it, that's basically a thin sheet of skin at the end of the outer ear canal, and that vibrates in response to sound. So here we see the first place where that physical energy is meeting your body. And, the same way that you'd have, let's say, I don't know, let's say you throw a stone in a pool and that creates vibrations, right? That's create waves. Those waves, let's say you're in the pool, they could reach, whatever, let's say, the end of the pool, right? And then you can feel those vibrations. So that's the first kind of point where the sound meets the body. And so yes, it won't leave you deaf, but it would basically, you could still possibly damage your eardrum. But let's talk about how the eardrum transforms those sounds into something that your brain can work with. So again, I'm talking about this part here. And maybe before I continue, let me actually share with you a very nice video, and then basically in the next slides, I'm going to clarify what that video is going to do a much better job of explaining. Okay.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch, or frequency, produce a slower rate of vibration. And sounds of lower volume, or amplitude, produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. Two structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the two fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
So later on, you can actually use that video and kind of unpack it, and that's what we'll do right now. But you can even pause it at different stages, and you can actually, go through the pages of the book and see how the seemingly abstract or the relatively dry writing can actually translate into this really neat, for in my taste at least, kind of audio visual kind of demonstration of the auditory system. Let me just kind of continue with the middle ear. Okay, so we talked about the tympanic membrane, and then we talk about the ossicles, which are those tiniest bones. They are the tiniest bones in your body, and the idea is that they amplify and kind of make sure, or they enable our ability to detect very, very faint sounds by amplifying those sounds to the inner ear. And so we have three of them: the malleus, the incus, and the stapes, and you can see them here. Okay, so here is the tympanic membrane. It rotates based on the pressure of those molecules, those sound waves we've been discussing, right? And then what you can see is that it generates this kind of piston-like kind of movement in the malleus, following with the incus, and then with the stapes. And the stapes itself hits, what we call here the oval window, or the base of the oval window, and that is going to create pressure, or we're going to call the pressure bulges in the cochlea. Okay, so quick recap. So we're talking about the middle ear, we're talking about the ossicles, which are those three bones, and they, as I've said, provide amplification. Okay, and by two means. One is just the fact that we have that hinge that I was telling you about, that piston. So that basically it's like a lever. So you can amplify that sound by actually to the 3 times more than the original sound. And then the stapes, by the fact that they have smaller surface than the malleus, so that energy is kind of concentrated. Okay, so that kind of difference in size creates basically concentration of that energy. And so basically you're going to have these kind of changes, okay, these kind of fluctuations in the liquid that's found inside the cochlea. Let me skip that real quick. So the cochlea is kind of the key part of the inner ear, and that's where the first part, or the first place where those acoustic vibrations are going to give rise to actual neural signal. So fine changes in sound pressure are going to be transduced into neural signals. And you can think about that in a kind of very coarse analogy to the retina. So the retina, what does it do? It translates changes in wavelength, right? Or intensity in terms of luminance, into different firing rates. Remember, we have the photoreceptors, we have the rods and the cones, and there's a kind of a cascade of events, right, leading to the firing of the ganglion cells. The ganglion cells, you can think about them as the first step in your optic pathway, the visual pathway. The same way you have here the auditory pathway, in the first pass, or the first kind of point where that is going to allow a neural signal to be fired is going to be at the cochlea. Okay, so you can think about the cochlea almost as like your retina, and within the retina we have the photoreceptors. Here we're going to have the hair cells. Okay, so cochlea is kind of like a spiral. It's a spiral structure of the inner ear, and that contains the organ of Corti, which you've seen previously. And let's kind of start to look at it in more detail. Okay, so here's your inner ear, here's your eardrum, and those three bones we were talking about. And then this kind of snail shape is the cochlea. And so if we kind of like unroll it, so to speak, you can see it here. I take that back. We're going to talk about unrolling it when we talk about representation of frequency. But if we kind of focus on it, if we remove all the other kind of parts, so first of all, we can see the part that we're not going to talk about, which is the vestibular organs. Remember, I told you that in the ear we also have the kind of maintaining balance system. What you have here is this cochlea here, and here's a section through the cochlea. Okay? And so imagine that you put like a knife or a very sharp kind of slicer, and you would have cut it like that. And then if you look at it, you kind of tip it over and look at it, it's going to look something like that. Okay, so here's a cross section of the cochlea. And basically, it's like, like two folded kind of, pipes, okay, or tunnels that have another tunnel within them, or a canal. So here you have the vestibular canal. Here you have the tympanic canal. In between, you have the middle canal. That's kind of, sandwiched between them. And you have these kind of two membranes, which I just want to give them names. So first of all, the vestibular canal extends from the oval window at the base of the cochlea all the way up to its apex. Okay, that's the canal that's closest to the ossicles. So the pressure waves go first, as you saw in the video, from here, from this vestibular canal. Okay, so it's going to make a trajectory basically in this direction. So from the vestibular canal all the way to the tympanic canal. Okay, and in the way it's going to basically create changes, or it's going to move this thing here, this purple thing, which is known as the organ of Corti. Okay, so we have the tympanic canal extending from basically, again, kind of sandwich, and it's at the base of the cochlea, and then the middle canal. The middle canal is sandwich, and the tympanic canal is at the bottom. Okay, so these are important names, and here are those two membranes I was telling you about. So there's Reisner's membrane, and there's the basilar membrane. And of the two, the most important one is the basilar membrane. It's not really a membrane, it's more like a plate of fibers, which are right here. Let me show you a picture of that. So this would be the basilar membrane right here. Here. So that's going to be your basilar membrane. Okay? And on top of it, you're going to see the organ of Corti. So here is the organ of Corti, structure on the basilar membrane of the cochlea that is composed of hair cells and dendrites of the auditory nerve fibers. So this is kind of the important part. And again, I think you're starting to understand why I started with the video, because there's just so many moving parts. And it's kind of interesting. The book calls it like a Rube Goldberg machine. So I don't know if you've ever heard the term of a Rube Goldberg machine, but it's kind of like those silly cartoon kind of things where, one action leads to another, which leads to another, and leads to another, and then there's a ball rolling, and then there's a liver falling, and whatever, and then it leads to some very simple or relatively rudimentary kind of action. So this is a series or a sequence of kind of, Rube Goldberg-esque kind of actions that would eventually lead us to fire, an action potential that would denote a certain sound is in the environment. So this together is known as the cochlear partition. And so this is basically the organ of Corti, and you can see on top of it is this kind of tectorial membrane. Okay? And long story short, what you have is this kind of, let's say, roof that's lying on top of this organ. And you can see that here you have these hair cells which are connected. And to those hair cells, you have the actual auditory nerve that's kind of in tune with them. So in the vision analogy, this would be your ganglion cells, okay? And your hair cell would be your photoreceptors. So long story short, your hair cells are going to move. They're going to move by this process of shearing between that tectorial membrane and that organ of Corti. And that movement, that shearing movement, is going to evoke a certain change in the actual potential of the hair cells, and that's going to evoke a firing in those auditory nerve cells. Okay, so there's going to be a synapse, okay, and that's going to evoke that communication between the hair cells and the auditory nerve. So hair cells and cells that support this thing called stereocilia or stereocilium in singular, and those transduce the mechanical movement in the cochlea into the neural activity that will be later on sent to the brainstem. And you can see them here. They're very, there's a nice kind of order fashion. So on top of these, those would be those cilia, like those individual quote-unquote cells or hair cells. You can see you have two types, the inner hair cells and the outer hair cells. And long story short, the inner cells, you can see them here, they're actually less numerous in number. There's about, I'm going to say, 3,500 compared to 10,500, outer hair cells, but the information kind of ascends or goes in an afferent fashion. So here you see the term afferent fibers through the inner hair cells. And later on, you're going to have some feedback, some efferent kind of information coming through and basically activate those outer hair cells. Okay? And here's this kind of description of how this tectorial membrane kind of moves along based on vibrations that are induced by sound. And importantly, kind of in the last 2 minutes, I'm just going to explain this notion of place code. And the idea would be that different parts of the cochlea, so from the base all the way to the apex, are going to be sensitive to different frequencies. Okay? So you're basically going to have something like that, or maybe I can just read it out first so you'll understand what I'm talking about. So place code is, or code, it's decoding of frequency. It's tuning of different parts of the cochlea to different frequencies, in which information about the particular frequency of an incoming sound wave is coded by the place along the cochlear partition with the greatest mechanical displacement. So that's a very, fancy way of showing you basically that if I unroll the cochlea, what you'll see is that different parts of the cochlea are going to be differentially sensitive to different frequencies. So high frequencies are going to displace, more the basilar membrane, so more at the start. Whereas the kind of tip, the apex of the cochlea, that's going to be most sensitive to those low frequencies which travel a longer way. Okay, so you can see it here, those traveling waves, kind of, in the liquid of the cochlea, right? And you're going to see that the closer you are to the base here, the basilar membrane, then you have the higher frequencies. In this case, let's say 1600 hertz. And as you kind of farther go away from the base, that's where you have more kind of flexibility in the cochlea, in the actual cochlea, and then you get sensitivity to the very low frequencies. Okay, so lower frequencies are farther away from the oval window because low frequency sounds have more energy, so they carry farther than high frequency sounds. So that's going to be basically the basic mechanism that would now lead to basically to a signal that's going to be sent through the inner hair cells. Okay, so they basically convey almost all the information about the sound waves to the brain. And then we have the auditory nerves, which are going to, I'm just going to skip here just because we've reached the end of the class, which are basically going to fire to the brainstem and then going up to the thalamus, to the medial geniculate nucleus, not the lateral like the envision, and that's going to send information all the way up to primary auditory cortex. So here is your medial geniculate nucleus, here is your primary auditory cortex, and this is where you're going to code the different frequencies and the different sound intensities into audible sounds.